Very nice to be here with you. And as Karel already mentioned, will be about Pharmacophore. So actually, the title is Pharmacophore Modeling, How to Enhance Virtual Screening Efficacy in Early Drug Discovery. And you are invited to download this presentation from this link here. So take a picture or just type it into your computer. And so afterwards you have the PDF of this presentation plus some additional material where you get some background information about the software that we will be using afterwards. So does anybody have the link?
Can I put it onto the Facebook?
Sure, yes, sure. Okay. So we have to, when we talk about all these in silico methods, we ask ourselves to get answers to important questions. And the important questions is basically can be divided into two big parts. When you do drug discovery, you try to find new molecules that can interact with a target. And normally there are two different workflows. The first workflow is to find a starting point. And to find a starting point, there can be some experimental methods, there can be some literature search, but there can be also some in silico stuff. And this is normally referred as to being virtual screening. And in virtual screening, you identify first starting points, nothing else. It's not that you will find a drug by virtual screening, right? You just find new compounds. And here the question is to optimize your selectivity towards the target and to exclude as much as possible false positives. False positives is a compound that you would predict to be active but is not active at the end when you test it. So imagine you have millions and millions of possibilities nowadays that you can buy compounds. There's something like probably 30 to 40 million compounds that you could buy. And if you want to test 10, 20, 30 of such compounds, you need to be really, really selective to retrieve out of this huge number, this small number of compounds. Then you have your starting point. And then you do hit to lead and lead optimization. And in lead optimization, you can also use these methods, but you have a completely different question that you answer here. So you want to optimize your structure towards multiple key properties. It's not only anymore the fit to a specific target, but you also want to avoid the fit to other targets. You want to have some physicochemical properties, some solubility. We had a very nice lecture this morning about that. So these are two different things, and it's not one solution fits all. So I will try to explain to you in which of these cases what methods can be used. So what is a pharmacophore? Actually, this is something that is nothing new. This is a pretty old concept that was already kind of envisaged or imagined by Paul Ehrlich in the early last century. This is the official definition from the UPAC. And it says it's an ensemble of steric and electronic features that is necessary that two molecules can interact with each other. So basically, it's a description of interactions. Right? And when you think about pharmacophores, you can think about features, interaction features, and these interaction features describe more or less the binding of two molecules to each other. And normally when we think about drugs, we always think about a small molecule, like a drug molecule, and some protein or DNA, RNA that this molecule will interact with. And so the interactions that are driving or the forces that are driving these interactions are electrostatic forces, H bonding, which is also a sort of electrostatic interaction. Then we have aromatic interactions, which is also some kind of electrostatic interactions. Then we have hydrophobic interactions, which are entropic interactions. We have coordination to metal ions. And this can be really derived pretty easily from structural biology methods, for instance, from X-ray crystallography of ligands co-crystallized with your target. You can immediately derive these interactions. You can store them like in three-dimensional space, and you can even, for communication, flatten them down into a sort of two-dimensional description where people can talk to each other. And it's very, very useful, especially in drug discovery, because drug discovery is not anymore a sort of single-person effort, but it's teamwork. And so when people can talk to each other, looking at the same thing from different angles, you will very often see that chemists love this kind of representation, while modelers love this kinds of representation, and biologists don't take any notice of structures anyway. They just know the name of the compound and the activity, and they don't care about it. But it's important that at least that chemists can interact with modelers. So, saying this, why am I in the Pharmacophore field? Basically, we have been using Pharmacophore since almost 30 years, and we were not happy with the methods that were out there at that time. And this is already something like almost 15 years ago, where we had all the different programs that were available commercially at that time. And we used all of them, and were not happy with the results that we obtained because they were different, actually. And so when we look here at the structure-based model, the same coordinates of the features, and we also use the same database, same conformations for screening in different platforms, in the Catalyst Discovery Studio platform at that time, in the Phase Schrödinger platform, and in the CCG MOA platform. We got with the same model, of course translated into the language of these different programs, but the coordinates of the features were the same, and the coordinates of the conformations were the same, but we got different hits. And the number of hits that are similar between the three different programs is low, and the overall number was really very low. So we were completely unhappy with that. And the same thing happened also when we used ligand-based models. So we generated one model with Catalyst here, and saved the Pharmacophore feature coordinates into the different programs, and did screening again with the same conformations, same compounds, and again the number of common hits was low. And so what came out actually is that these different programs, even if they are fed with the same conformations and the same Pharmacophore coordinates, they would align the compounds differently to the Pharmacophore models. And they would have different kinds of functions, all basically graph matching. And they were pretty slow, and the more features you had, the slower they would get. So people would try to keep small Pharmacophore models with just 3 features, be happy with that, but this was not really good. And so we decided to do it in another way. So it turned out that these old Pharmacophore methods suffer from these limitations. Different tools return inconsistent results, as I told you. Alignment by graph matching was slow, and we had to use low number of features to be somehow fast enough. And this means that you're inaccurate. So if you have a low number of feature, I mean, you will find a lot of false positives. And so the solution is we sat down and rethink the problem. We decided we have to redesign the alignment algorithm. We need to avoid this computationally inefficient graph matching. And we decided at the end to create a pattern recognition-based approach to do Pharmacophore comparison to each other. And so what we do is actually work in the Pharmacophore space. So we transform every conformation of every molecule into a Pharmacophore model. And when we screen with pattern recognition this Pharmacophore model of each conformation to the given Pharmacophore model. And so what we did is this pattern recognition. Why is this so powerful? Is because pattern recognition is a fuzzy technique where you can read this easily, right? So we use pattern recognition every second in our life we do pattern recognition. Even here, right? It's pretty easy to read this for you if you understand why. So the same thing is true for Pharmacophore models. It even works when you mix up letters and numbers. But you need to have a certain shape similarity between the letters and the numbers. So here you see, for instance. So it's easy. For us, we are pattern recognition machines. And so we said, okay, we need to use this same technique for Pharmacophore comparison. And this is what we did, actually. So what we did is we create, we know what the feature description is. So we know a feature is, for instance, a hydrophobic feature, or an H-bond donor, H-bond acceptor, etc. Then we look into the neighborhood of these features. We create concentric spheres around each feature, and we count how many times another feature occurs in these concentric spheres. And then we have the neighborhood of the feature encoded into the feature definition. So we know it's a hydrophobic that has an H-bond donor and an H-bond acceptor in a certain distance to it, and another hydrophobic, etc., etc. So we create like this typed, so-called type distance shells. And then when we look at these models here, these two models, try to find out what the common things are. I mean, this is for us, again, it's simple because we are already trained and we recognize this pattern immediately. And using this pattern matching algorithm, our software also recognizes that these are the two hydrophobics that match to each other. These are the donor, acceptor, donor pattern that matches. And this is already enough to make the alignment, because normally with 3 features you could do an alignment. Here we have 5 already. So the rest is just Kapsch rotation, translation, and now we know that this is the alignment of these 2 pharmacophores. And behind this could be 2 molecules, or it could be 1 molecule and a pharmacophore model that we want to compare with, right? And what we learn here is that we don't need the same number of features for comparison. We need just a few basic features that have the same type and the same neighborhood, and then it's enough. So 1 molecule can have 20 features, and the pharmacophore that we want to compare needs maybe only 7 features, but we will find the best solution using this algorithm, and pretty fast. And we learn something. Now, if these two pharmacophore models came from 2 molecules, for instance, what do we learn? We learn that 1 model has an additional hydrophobic group here that the other does not have. So we could think about to make this molecule better to add this pharmacophore element there. So we just need to put some more hydrophobicity here into the molecule. Then we see that here there is a region that where an H-bond donor and an H-bond acceptor comes together. So for us, this is clear. There must be an atom that can do both at this position. And there's another lonely H-bond acceptor here, where 1 molecule had an H-bond acceptor, the other not. So if we want to transfer this information, we can also transfer it to the other. So the great thing is that chemists can very easily translate this into structures and just think how they would need to modify their molecule to make it better, to fit better to a pharmacophore model. But also the other way around, how to avoid a certain pharmacophore that you don't want to hit. Because imagine you have your molecule and a lot of targets that you don't want to hit. All anti-targets that are related to toxicity, to metabolism, any kind of things that you want to avoid, you can just model as a pharmacophore model and try to avoid all these features that the molecule have, or modify your molecule so that it does not have these features anymore. So actually this is really a promising approach. And so together with Gerhard Wolber here, who was at the University of Innsbruck, where I was also at that time. By the way, he is now at the University of Berlin, professor. In 2004, at the EuroQSAR meeting in Istanbul, we presented this first prototype that we had created. And in a lecture that was called High Throughput Pharmacophore Model Generation from Ligand-Target Complexes as a Basis for Activity Profiling. So this was our first idea, to create as many models as possible from the literature using crystal structure data, basically, and parsing the entire PDB database and create pharmacophore models for each of these complexes, and then have a tool that can run in parallel a molecule against all pharmacophores, and this would give you a sort of activity fingerprint, affinity profile, right? And so we had called this Ligand Scout. This was the prototype here that was elaborated in 2003 in my group here in Innsbruck. And you would have a slider here where you could just go left and right, and when you stop, you would just get to any kind of PDB structure that would be there, and you could go stepwise, and then you would see all the complexes here already interpreted with the ligand. So there was a lot of work to be done, because in the PDB, as you know, There's no information about protons. There's no information about atom types. So you need to guess which atom type you have, basically, from the structure, from the 3D geometry, and from the interactions that this structure does with the protein. And so what we did is, and this was the Ligand Scout prototype, so we named it Ligand Scout, because we are scouting for ligands that are in a protein. And this software was just done to create structure-based pharmacophore models automatically on the fly. And then after we had done this, we decided, okay, now we have something very nice. Actually, we will implement this into a software that can be used in general and that can be licensed out to people. And we created a company for that. And so we then really had an evolution. So Intelligent, the company who I've co-founded together with Gerd Wolber and another professor, is actually currently still developing this software and marketing this software. And it went from automatic structure-based pharmacophores to the development of this alignment algorithm. Then in collaboration with Sanofi, we did ligand-based pharmacophore generation, clustering, and virtual screening. And then this was then version 3 was stopped here. Then we did a complete refactoring of the code, because up to version 3, Ligand Scout 3 was a monolithic code. So it was everything in one big piece of code, which is impossible to maintain anymore. So we decided to stop and to refactor the whole code. So the surface would still stay the same, but behind we reworked it completely, which took almost 2 years actually. And then there was the implementation of dynamic relational databases behind. Docking was and rescoring technology was implemented. We created KNIME extensions, because do you know KNIME, the software KNIME? This is something really, the ones who don't know it should look at it. Can download this. It's a workflow software that you can use basically for free. And there's a lot of community algorithms out there, and also from professional software developers like ours or Schrödinger's, Mo, etc. have nodes for this. And then the last thing that we implemented is pharmacophore-based analysis of MD trajectories. So the lecture this morning is very inspiring. And I guess what you could do is you could take the analysis of these trajectories to the next level and make it much more easy. And I will show you in the afternoon some examples how to do this. Because we go away from I would say, measuring of distances between atoms into a description of the interactions on a much lower level. So an H bond will be there if the distance is there, but it can also be moving from one residue to the other, but it still will be there. And sometimes if you just look at this, you will not see it anymore. Okay, so this is just a snapshot here from the surface, how it looks like. Basically, we will work with this, and you will see a lot this afternoon how this works. You can, for structure-based modeling, you type in here the 4-letter code, and it will automatically download from the PDB the structure. It will recognize the ligand here is a yellow box. While you zoom in, it will automatically attribute the atom types and bond types. And then you can just press a button here, and it will generate the interaction pharmacophore. That's it. And then you can take this pharmacophore and use it for screening or using for other kind of things that you want to do, comparison, etc. So I'm very happy, actually, that people are really using the software. And I just looked up yesterday how many papers have been citing our software. Is more than 1,700 at the moment. Since it's out, it's all about also structure-based modeling, ligand-based modeling, virtual screening. We have hit identification. We have fragment-based screening. We have lead structure optimization, protein-protein interactions, drug repurposing, and profiling even for side effects. You name it. Actually, it's really used worldwide in industry, in academia, in small companies. So it's really a tool that has been adopted widely by the scientific community, and I'm very happy about this. I just want to show you a few examples here, a few success stories from different labs, not from my lab here, but from different labs. So 2 years ago, there was just a story that they found the first dual inhibitor of a 5-lipoxygenase activating protein and of the soluble epoxide hydrolase, by pharmacophore-based virtual screening. So they were looking for this compound that act on two different levels, but in the same pathway, on two different systems. And they created pharmacophore models for both targets, and they found compounds that fit both targets. And then they measured and found really compounds that were on one target 20 nanomolars, in the other one 200 nanomolar. So this is not a drug, but this is a perfect starting point for optimization. If you have already something that is 20 nanomolar, you're pretty good. You just need to optimize a little bit with respect maybe to solubility or metabolic stability, but basically the potency is there already. And in 200 nanomolar, okay, you can just try to get it 10 times better. So this has been reported in Nature Science Reports here last 2 years ago. This was drug discovery for anti-inflammatory drug starting points. But also in agrochemical industry, it could be interesting to find compounds that would can be used for inhibiting insects to do certain things. And this is a Chinese work here that is also published in Nature Science Reports here, 2016, where they looked for compounds that would inhibit moths, these little nasty animals here, to attack fruits and then to destroy these fruits, basically. And what they found is, and they published a structure-based discovery of potentially active semiochemicals for Sedia pulmonella. This is this moth here. And they looked for compounds that would distract these animals. So by trying to finding pheromone kind of compounds that would just avoid these animals to attack the apples. And this was the first use of pharmacophore model for the discovery of these semiochemicals, actually. So what they did, a very, very elaborated approach. They did homology modeling for the target, and they did some docking of some known compounds, that they created the pharmacophore here that was pretty, I would say, a little bit strange. A big hydrophobic part on one side, then a lot of space in between, and the crucial H-bond acceptor here on another side of the molecule, and a shape limit. And they came out with compounds, they tested the compounds, and they were active, actually. So this was really a nice success story. You can just read it yourself. Another one, a very good approach also in 2016 that was published by a group in France, was discriminating agonist and antagonist activity. Even doing this, when you have, and this is something that has been under long discussion, if you can predict functionality by pharmacophore models. And most of the people would not believe that this works, but if you are selective enough, and if you have a lot of features and very carefully placed features, you can do it. And these guys here showed that it works, actually. They did a mix of structure-based and ligand-based approach and could discriminate between nuclear receptor agonist ligands and nuclear receptor antagonist ligands. So they created a whole bunch of different models and screened in parallel. And then according to the profile, they could say, this will be an agonist, this will be an antagonist, and it works. And then this is a paper already a long time ago, just after we had published virtual screening algorithms. A group in France here did a comparison of a lot of different virtual screening tools in order to evaluate the potential of these tools to correctly retrieve a certain class of molecules. And in this case, they were interested for CXCR4 antagonists, and this is a class of compounds that you would like to develop if you want to find new HIV drugs. Actually, this CXCR4 receptor is a host receptor that the virus needs to attack the T cells. And if you can hide this receptor by finding kind of antagonists in this case, then you would also prevent HIV to attack the cells. And this is one of the concepts that is currently pursued. And they had a very nice database of compounds that they had tested already. And when I saw the paper, I was not looking for that, but I just saw this, I read the magazine, and I saw this picture here, and then I read the paper. It's about this comparison. And actually, they created this structure-based pharmacophore model from one of the X-ray structures. It's 3OE6. And they show a very nice ROC curve. So are you familiar with the concept of ROC curves? Okay, we will learn this afternoon again. But basically, it's a sort of matrix for how well any kind of decision tool is able to discriminate between true actives and true inactives. And normally when you have no discrimination, you would get a line that goes here, because here you have the specificity and here you have the sensitivity. So here means 100% of the actives in the database, 100% of the actives retrieved. So what you want is something that is very steep in the beginning and not something that is like here, because this would be just as random, a random selection. And if the curve is below this line, then it would be even worse than random, right? So you would enrich inactives in comparison to actives. And so the curve that they show here is the Pharmacophore curve from Ligand Scout, and it stops here and then it's prolonged to this point by just creating a line here, because our screening algorithm will not retrieve compounds if they don't fit. So there will be a score of 0 given to that. And to make a comparison, because if you do docking, for instance, you would get a score for every compound. So we just make a line here then as a random selection line from here to here. And so then this is the original diagram that is published in the paper. And you see, in fact, that there are some methods that work lower than random. You can read it, which one it is. There are some that are a little bit better than random at the beginning and then worse than random. There are some that are a little bit better than random. And then there are some that are pretty good and they stop here. But still the best that they found was the Pharmacophore method using the Ligand Scout Pharmacophore that does not retrieve all compounds, but retrieves like 87% of the actives in the first 5% of the molecules screened. So you get an incredible enrichment, actually, and this is what you want. You don't want to screen experimentally all your library to find all the actives. You want to screen a small fraction of your library experimentally to retrieve most of your actives, or at least a fraction of the actives, right? But not inactives, so this is what you want to do. And so the people here, they stated that the Pharmacophore that they developed is highly specific and sensitive, and is able to retrieve a diverse scaffold pool. And of course, it's a special case. It does not need to work in all the cases the same. I completely agree to that. But in general, if you do it well, a Pharmacophore is at least in comparison to docking, it will perform at least in the same way. But the time will be much, much, much faster. And if then you can really filter out a lot of compounds that you don't want to further investigate, and you can focus on a small number of compounds that you want to investigate. So what can you do here? You can do, for instance, fragment-based design. And this is one story that where I was a little bit involved also here in the design of the study. We just published this also a few years ago already. This was at the time when I was heading the Prestwick Chemical Company, which is a research organization located in France. They are very good chemists, actually, excellent chemists. They put several compounds into clinics. One is on the market. And we had one of our customers had a pretty difficult target. This is the protein. And actually, there was a public structure of this protein with a peptide. It's a protein-peptide interface, basically. And they wanted to find compounds against this target. They could not do any kind of animal testing, so they are completely forbidden to do animal testing in this region, in this field where they are. And so they wanted to do something using the chemical space that they know is safe. And this is something like 290 compounds or so. So with 290 compounds, you will never find anything in this space here. So our idea was then to take these compounds that are considered as being safe and cut them into pieces and recombine them. Because our idea is, I mean, if you take a safe molecule and you recombine it in a way that makes sense from a chemical point of view, the chance that you get something that is all of a sudden toxic is low. I would say. I mean, you can discuss it if it's a right assumption or not. I mean, I just say the chance is low. And they bought into this concept, and finally what we did is we did a fragment-based screening. So we did not use the whole pharmacophore for screening, but we chopped up their library of compounds into small fragments, and then we looked into each of these space of the pharmacophore if there would be a fragment that would fit into there, and then we would combine these fragments and synthesize the molecules, and then do the testing. So what we did is we used these smart fragments, etc., etc., and when we synthesized these higher-strength solutions, we looked, of course, for IP situations, so we don't want to synthesize something that is already existing, of course. It needs to be new and patentable. It needs to fit the target. It needs to be easy, so we don't want to have like 20-step synthesis. We want to have something where the chemical tractability is good, and of course the physicochemical properties that we can calculate should be good too. And so what we had is 274 structures initially that they gave to us. We chopped them up into 837 fragments. This was done manually. This was a medicinal chemist. For 1 month, he looked at all molecules and cut them in pieces at places where you could use a chemical reaction to recombine. So, for instance, an amide, which would be very clear, or ring closures, etc. So we had 837 initial fragments, but a lot of them were duplicates. So we had at the end 582 unique fragments. And then to make it a little bit faster, we created a library of 91,000 compounds by combining these 582 fragments by 582. So there are some that you cannot combine because there are reactions you cannot do, but we got at the end 91,000 fragments. Like still small molecules that have a molecular weight of below 300, etc. So the typical fragment-based thing, molecular weight lower than 300, a maximum number of 3 for H-bond donors, maximum number of 6 for H-bond acceptors, and 3 rotatable bonds. This is the rules for fragments in drug discovery. And so Ligand Scout then, the virtual screening, delivered a reasonable small number of hits, so in the 0.005% range on these fragment pharmacophores. And then we looked into 2 fragments and combined them again and did synthesis biological testing. And so I cannot show you structures because we never published the structures. They are under patent filing at the moment. But the timeline is really fast. So one guy did the work on computer-aided design, and then he delivered the hit list after 1 month, and then 2 people delivered the first hits after 1 month, and we sent the hits off to our customer, and they did the testing, and they came back with something where I say, this is incredible. All the compounds that you sent to us are active, and they're in the low micromolar range. So it's not really highly active, but as a starting point, low micromolar range is pretty good. And so we started here from a hit to lead approach. And 4 months later, because we had this structure, the X-ray structure, we could very well design into the X-ray structure new molecules. And so after 4 more months, we could nominate the lead. But the best thing is here that the first hits had already novel structures. So even the hits were not described in literature before. And this is a really good situation because you just can do synthesis and you can be sure that nobody else is working on this field. And at the moment, they are in development, these compounds. So this is really great. So I think as a first summary, we can say that these pharmacophores are universal and a very rapid method for accurate feature-based generation is there. That we have highly selective models that can be used for retrieving low number of false positives, therefore a high enrichment factor. The question is where to use this in the drug discovery pipeline. I think this is pretty clear. It needs to be used in hit identification, right? So this is really a good way to start hit identification, but what are the next steps to integrate? And here I want to follow up exactly the talk that was in the morning and just look at this picture here. This is a nice picture of a bird here flying over this monastery. It's in Italy, actually, and there's a very nice conference at this place in Siena. the workshop in drug discovery will be this year again. And this is another picture here of these birds, and another one. So it's pretty difficult to get these kind of pictures. It's not easy, right? It's sitting up there somewhere in the high altitude and being lucky to have these pictures here. But you can do this, actually. You can take a video and then take the snapshots out of the picture. And this is exactly what we do when we do MD simulations. And molecular dynamics simulations are not only interesting now anymore for academic people who have a lot of time, but they also are interesting now for industry because of computer power getting more and more available. And especially this parallel computing hardware in GPUs. You know, you have these graphic processors where you can run a lot of processes for MD simulations on a graphic card at the moment, which is no longer in the CPU. And so now this becomes available. So you can have clusters of GPUs. You can run simulations. You can run long-term simulations. Still, analysis of these trajectories is still something that is long and cumbersome, and to find out what's coming out. So the interpretation is difficult, but pharmacophores are a perfect solution for that, actually. And I just want to draw your attention to this review here from Jérémy Mortier here, this article here. The guy, he's at Bayer now, and he writes about the impact of MD on drug design. So there's really lots of interesting examples that you can find where MD simulations were really crucial to find new compounds in drug discovery, in industrial drug discovery. So the question is now, when we analyze these MD trajectories, how to find the relevant pharmacophores, which ways shall we use to calculate similarities between these pharmacophores. We need to be aware that we need to identify rare events that might be important ones. And use these models actually as a way to abstract the ligand-protein interactions in these MD trajectories. And so what we did is, first we implemented this method in Ligand Scout so that you can load a trajectory in Ligand Scout, run it, generate all the pharmacophore models of this trajectory, and analyze these models. So this was done. And we did a first proof of concept study with a data set of 40 protein ligand complexes selected from the PDB DUDE set. So where we restricted us to single ligands, no metal ions, and less than 200,000 atoms. We did large MD simulations, 10 times 20 nanoseconds with charm, and saved coordinates every 10 picoseconds. And then we generated for each of these frames a pharmacophore model. So we had like 10 times 2,000 coordinate sets, 10 times the PDB structure. So for each of these 40 proteins, we had something like 20,000 pharmacophore models. And this is, of course, way too much to analyze in a graphical way. So we converted these pharmacophores into pharmacophore vectors, basically, and did a feature frequency analysis in these vectors. And then, because we saw, this is just the different target families that we were used, so sort of more or less a representation of what is representative in the PDB. And the way we encoded is actually for each atom of each molecule, there is a potential pharmacophore interactions if there is a partner in the protein. And so what we look at is which atom undergoes which interaction and how many times of the trajectory is it there. Is it there at the beginning? Is it already there at the PDB structure? Or does it come up later, etc.? And so we can create a sort of vector here where each line, so these are all the vectors of the trajectory underneath each other, and we can rank them according to the number of occurrences. So this first model here would occur 1,800 times out of the 20,000 times, so this would be 9% approximately. And you can see which are the features that are occurring. Are they already the same as in the PDB model, or are they only in the MD? And this is the initial pharmacophore. And so what we see is that the initial pharmacophore here occurs only 43 times out of the 20,000 times. So this means that this is not really something that is relevant. So we really learned something, that this initial pharmacophore model from the PDB structure might not really be the best for doing the screening and for predicting activity. Also, the highest frequency model does not necessarily be the one that gives the most information. So what we did is then we looked at all the possibilities. So in a typical case, out of these 40 proteins, we have something like 260,000 possibilities of potential combinations of pharmacophore features when you think about all potential combinations. But in reality, we only found 50 to 400 combinations really existing, depending on the target. So already this gives us some information that some combinations of features will not exist. So therefore, we should not have these combinations in molecules that we want to synthesize, because they make no sense, right? What we also learned is that many features are not present in the initial PDB-derived pharmacophore model. This was a little bit disturbing. And also that the initial PDB model does not have the highest frequency, which is logic somehow, because when you create a crystal, it's something static, and it's a kind of crystal under certain conditions. So it's energetically low, of course, but still, I mean, you do some artificial things to that, some packing, etc., etc. So probably this is why this doesn't have a high frequency. And it also turned out, because we did virtual screening with each of these models and looked at the enrichment factors and the retrieval rates. And so, unfortunately, what we found is that frequency does not seem to have an impact on the quality of the screening results. So what I mean with quality of the screening result is how well a model can discriminate between true actives and false positives, right? I don't want a model that retrieves a lot of molecules that are inactive. But the high-frequent models are not the ones that do this, unfortunately. So how to find really the best model in this case? How to use all this information? So then we came up with an idea to do this in a double step, actually, using a post-screening process, using the underlying principle that a true active molecule should have a higher chance to fit a number of pharmacophore models derived from these dynamics than an inactive one, who would maybe fit a certain number, but surely not all of them. And so what we do is, if we do a consensus fit, using all the representative pharmacophore models that occur with a certain frequency, we should be able to get a better enrichment. And so what we do, we called this approach the so-called common hits approach, CHA. First, we derive all the hits from all the models, and then the hits are ranked by the number of models they fit. So a hit that will be retrieved by a lot of pharmacophore models will be ranked high, and the molecule that is only fit by a few pharmacophores will be ranked low. And so then we compared these enrichment factors, and here you have the different matrices here at the different percentage in the area under the curve. And please have a look first at the PDB model. So the PDB model works pretty well until a sort of enrichment up to 20%. Then it drops drastically. All the others drop also. And then when we do the common hits approach performs perfectly up to 20%, and only when you go down to 50%, it drops. But we don't want to screen 50% of our database. So if we can retrieve most of or almost all of the actives in 20%, that's fine. We are very happy with that. It's much better than the other methods. So the area under the curve is still close to 1, whereas with the PDB model, it's already something 8.5. With the high-frequency model, it's around 0.8, and with the it's even lower. So basically what we know is now this common hits approach is really working well in boosting the enrichment and boosting the area under the curve up to 20%. Then it becomes worse. And also then we looked at the, so this has been published actually. This is a paper that is published already in JKIM. It's called the common hits approach. And there should be a reference here somewhere, but I'm sure you can find it. This is not yet published, but then we did the analysis of how similar are these hits to the original structure, because it's always interesting not to find very similar compounds, but also dissimilar compounds that have the same pharmacophore interactions. And so what we did is we did a clustering of the entire hit library here and look and make a projection here on a self-organizing network. And we know exactly where the original compound is situated, and then how far the hits are means how similar these hits are to the original hit. And so we can then compare also the structures in this dendrogram here on the left side, hierarchical clustering, to the projection of the into this self-organizing network. So when we look here at the first 0.1% of the database screened, you can here see the common hits approach, retrieved hits, and here the hits that are retrieved from the PDB model. And the initial ligand is situated here. So you see that in the first 0.1%, more or less, the PDB pharmacophore is more or less giving the same results as the common hits approach. The common hits approach gives a little better coverage here. It finds this molecule here, this one, etc. So there is a small improvement. When you do this with 0.5%, you see that there is also compounds already retrieved that are far off here. For instance, this one, also here there is one compound, this gets bigger, and coverage here becomes better, but here it only retrieves this one and not this one, for instance. So coverage becomes much better here. Same thing when you go to 1%, etc. So you can see here the similarity of hits, even when you go to a higher portion of the database screens, remains closer to the original hit when you do the PDB pharmacophore, but it gets much better when you do this common hits approach. And at 5%, you see you really cover a lot of different space, where here in the PDB you still are in the original ligand space, right? With one exception here. I want to skip this one, but the diversity here, the number of retrieved clusters for common hits approach and PDB model, at the beginning it stays the same, and then it separates each other, and the PDB stops at something like 40%, whereas the common hits approach reaches almost 65% with of hits that are structurally diverse. So actually, what we still need to continue to work on that. A growing number of trajectories is available. It's difficult to interpret them from design guidance, so we need to easy understandable interaction schemes. And the first steps will be to focus on specific regions to look for water-mediated ligand-protein interactions. This is something that is very important. Water in dynamic simulations, of course, correctly set up. Is this a water molecule that is entropically favored or entropically disfavored? Does it want to be replaced, or does it want to stay there? And so what we do is we are currently working on pharmacophore feature frequency analysis for prioritizing replacement of molecular substructures and water molecules especially. So this is what you will see later on. We will generate these pharmacophore models from a trajectory, and you will see the dynamic appearance of these models coming and going. And we will do an analysis of such models, looking at high-frequency pharmacophores, filtering down all the ones that are less interesting, looking at a specific feature, showing where in which of these models this feature is occurring, etc., etc., looking at all the pharmacophores together, saving them, and so on and so on. So what we are currently working on is now to step one step back and even analyze these pockets originating from trajectories without having a ligand inside. because this is what you want to do. You want to look at your protein, and maybe you put a ligand inside, and then there's a pocket opening somewhere else. And this is a pocket that you want to attack, for instance. So what we do is we set up a method where we perform grid calculations. Basically what we did already on a static, we now transferred into dynamics, where we can define buriedness and druggability thresholds, and we can calculate interaction probabilities on each of these grid points according to our heuristic rules for the pharmacophores. And so we can say at this grid point there's a high interaction possibility that this can form an H bond, for instance, to this amino acid in the protein. So we need to align these grids, of course, and we need to analyze them and look for emerging binding pockets, especially hot spots for interactions, and water. Water is extremely important in this case. And I want to show you one example here, coming from a PhD thesis of one of our students here who was working in collaboration with people. And this is water molecules here. So these are the so-called unhappy water molecules. Means that they are entropically disfavored. And we can calculate this, actually, and also look at the happy water molecules, which will appear just in the next part of this movie. These are water molecules that where you constantly see interactions with protein or ligand parts. And what you see is that there is here a little position that will always be there. So there will always be water there and will always be a happy water molecule. So this is a water molecule that you should not replace, right? And unhappy water molecules are water molecules that you should replace. So this is exactly what comes out of such an analysis. And so actually this can be really used then in lead structure optimization. So you really get easy to understand with design guidance. You can focus on specific regions, and I call this unhappy and happy. And this is, of course, stupid to say it, but it's easy to remember. And of course, it has nothing to do with happiness. It's just thermodynamics. But I think that for you, it's understandable. And for instance, these entropically disfavored, unhappy water molecules shall be replaced with small hydrophobic substituents. And this is the typical thing for the magic methyl. So when you are in lead structure optimization, at the very end, sometimes you really do a sort of methyl screening. You try every position of your molecule to put a methyl group. And sometimes you are lucky and you find a position where this methyl will just boost the activity of your molecule. But when you do this in a rational way, you can just not do all the other molecules that you would need to try. You just make the right one. And so you get the really idea for where to position this magic methyl position. So using this Pharmacophore hotspot feature analysis, you can really prioritize replacement and modifications of molecular substructures. You can provide interaction preference guidance, and this can be also easily adaptable for automatization of design of molecules, de novo design. And this is also something we are currently working on, using artificial intelligence to create molecules that fit into this dynamic pharmacophore. So it's pretty a lot of things going on at the moment. And I want to finish with a few examples here where we did this analysis, this pharmacophore analysis of trajectory analysis, to reveal predominant ligand-protein interactions, for instance, with analogs of modafinil. So this is modafinil. This is a drug that has been developed and approved in the US against narcolepsy, so people that fall asleep just during the day, they can stay awake with this. It's an inhibitor of the dopamine transporter. And I have heard that students take this to be better in learning, so it's a misuse. And the US Army is using it for their pilots when they do long-term missions, so they get this and they stay awake for 24 hours, which is crazy, and they stay really conscious and awake. So what we did is we analyzed these compounds here in two different isomers, because this sulfur is chiral here, and there's an S and an R configuration. And we can see predominant ligand-protein interactions with this amino acid here, really nicely shown in one case and not in the other. And also another one with this tyrosine, also in one case and not the other. And so we can explain why one enantiomer is much better than the other one. And we tested it, and it's really true. And based on that, we derived a lot of analogs and could synthesize molecules that are way better now than modafinil and working also in animal models. Another example is analogs of valerianic acid. Actually, this is this compound here, the acid and also the amide, is a strong GABA receptor modulator and subtype selective. So the GABA receptor is an ion channel that is built up out of 5 units, and they are called alpha and beta units, and so there are beta 2, beta 3 subunits selective, and this is related to anticonvulsant and anxiolytic activity actually. And of course, you can imagine that this is a nightmare to synthesize. I mean, this is a natural product. You cannot patent it because it's a natural product. And so to make synthetic modifications of this molecule is really a nightmare. So we were looking for much simpler molecules. And we did a lot of studies here, docking, MD simulations, pharmacophore modeling, and this is basically what we found, doing also mutagenesis studies, really to understand which are the amino acids who interact with this valerianic acid here. And we know that there is H-bond acceptor features and hydrophobic features and the shape. It's really a small molecule that you can see here enlarged a little bit. So we have a pretty good idea about the interactions, and so we did some design and synthesis, and when you compare the overall yield of the valerianic acid, 13 steps, we found compounds in 2 to 3 steps with an overall yield of 80%. We patented this and just was published early this year, came out. And the interesting result is that they are way better, smaller, simpler, and better than valerianic acid. Both on the showing the Vmax here, Emax, and also the elevation of Caesar threshold in mice compared to the, so this is the valerianic acid here, and this is at a much lower concentration. You get a higher affinity, higher activity in vivo for avoiding seizures. So as anticonvulsant. So I want to conclude here this lecture with these few key messages here. I think that this pattern recognition-based technique is really superior to all previous pharmacophore methods with respect to speed and accuracy. And you will see yourself then when we do the training how fast this method works when you screen libraries. So this is highly useful for hit identification. I think what we wanted to learn from this dynamic story is how, and we still don't know it, but we will finally, I guess, to know when you have a certain target, is it necessary to do a simulation or not? Is the PDB model enough, or do I need to do this calculation? So I think that we will know pretty soon what kind of feature distribution you need to have a very good model at the beginning and not to do the dynamics. So it is highly useful for hit identification, but we can transform these static models into something dynamic in a convenient dynamic approach, which is highly useful for least structure optimization, of course, because you get really the whole picture, which part of your molecule you need to modify, especially when you look at these water molecules, for instance, and where you need to put more interactions. And so, of course, there's a blend here. You can, of course, using this method also for hit identification, but you can imagine that this is a lot of more calculation effort than just pressing a button, generating a model, and screening, right? Sometimes it's necessary to do it. Sometimes it's enough to do it like that. And so the common hits approach retrieves more structurally diverse molecules than the PDB. And the higher the number of considered molecules, the more diverse the retrieved hits. And we are still working on pharmacophore fingerprints methods to do the comparison. And so with that, I guess I'm at the end of my introduction to pharmacophores, and I want to thank you for your attention. Yes, it's